good morning. Hello to all of you, and thank you so much for joining us. This is Susanna Rancevaso. You can also see my colleague, Dr. Jenny Almaco. This is EURAXESS ASEAN, and you are joining us today for a Meet My Lab session. Now, what is Meet My Lab? Meet My Lab is a virtual networking platform. It is a platform for researchers with similar interests that are located in different parts of the world. In each session, we are inviting a researcher or a research team, and we're giving them the opportunity to present their work to their peers that are based in Europe, in Southeast Asia, anywhere across the globe, and giving our audience the opportunity to ask questions, to look for linkages, and to hopefully find common ground for collaboration. In this session today, we're starting with a very interesting talk. And our very first speaker is Dr. Nicholas Kurniawan. You can probably tell from his name, he is from Indonesia, but he is joining us today from the Netherlands. Nicholas is currently assistant professor at the Department of Biomedical Engineering in Eindhoven University of Technology, but he started his academic career in Indonesia. He later on went to Singapore, where he studied for his PhD. He holds a PhD from the National University of Singapore, and afterwards it took him to Europe as an MSCA fellow, a Marie Curie fellow in Amsterdam. He's talking about his research to us today, and his current research focuses on understanding how the physical and the mechanical interactions between cells and cellular environments shape physiological tissue function and drive pathologies. His interdisciplinary team combines approaches from biophysics, soft matter, mechanobiology, microfabrication, and cell biology with an outlook of exploiting cell materials interactions for biomedical interactions. So really a very heavy, very interesting topic. Nicholas has received funding from various funding agencies, including the European Research Council. He's the holder of a European Research Council starting grant, which he obtained in 2019. But his work is also funded by the Dutch Research Council, by Health Holland, by Holland High Tech, as well as a national consortia called Materials Driven Regeneration. Now, Nicholas is also the associate editor for BMC Research Notes, which is a Springer Nature publication, and he's an editorial board member of the journal Communications Biology. Once again, this is a Nature research publication. As I said, Nicholas is originally from Indonesia, and he is a very dedicated member still of the Indonesian research community. He serves currently as the Deputy Managing Director for Europe for the International Indonesian Scholars Association. Now, before I invite Nicholas to start his presentation, I just want to quickly say a few words about housekeeping. We're hoping for this to be a very interactive session. So Nicholas will in a minute present his research to us, and then it's up to you to ask your questions to him. Please keep your microphones and your cameras switched off. When you do want to raise a question, please send us a message in the chat box. We will create a list, and once it is your turn, your microphone and camera will be enabled, and you can pose your question directly to Nicholas after he has finished his presentation. Now, I think with this, Nicholas, it's your turn now to dazzle us with your research and to introduce your lab to us. Thank you.
All right, so so good morning to people who are in Europe and good afternoon, I guess, for people in Southeast Asia. Thanks for the introduction, Susan. Let me try to share my screen. All right, so I hope you guys are seeing my slide, my PowerPoint.
We do, yes.
Great. Yeah, so you know, when Susanna approached me to join this event, you know, Meet My Lab, the first thing that came to my mind was like, you know, this is a Corona situation. I'm not able to actually bring you guys into the lab and meet my lab members and, you know, group members and what kind of research we are doing. So, which is a pity, of course, but that's how it is right now. So what I thought I would do in the coming, you know, half an hour or so is I will sort of give you a glimpse, overall glimpse, you know, just an overview of what kind of research we are doing, what kind of yeah, approaches that we are taking, and also who in my group is doing what. So in a way, it's kind of a virtual Meet My Lab kind of session. So I hope you guys are going to enjoy it. So let's start very general and start with thinking about cell biology in general. Now, the first thing that comes to your mind, I guess, when you think about cells is you know, the biochemistry of it. So biochemistry and molecular biology, and this is the traditional way of looking at cell biology. And this is the, you know, the first thing that you will see if you open up a book on cells, molecular biology of the cells, for example. So a lot of schemes about signaling, molecular signaling inside the cells, um, the central dogma, you know, stuff like that. Very biochemistry oriented. Now, I want to actually contrast that with what is happening in reality. And what I mean by reality is, you know, experiments in the lab where cells are cultured on culture flasks or some kind of a substrate. So that's kind of the first contrast. And the second contrast is what is happening in the body. And in the body, cells are really in a three-dimensional environment, and they're interacting with all kind of matrices, extracellular matrices with a variety of morphologies. Can be a sheet, can be fibrous network. So, and cells basically have to navigate through these physical environments, right? So physical environments that can physically interact with them. So this is something that can actually influence the way that the cells behave. This physical environment can also affect, you know, cell fate and a variety of behaviors that cells do. So I really like this quote from Mina Bissell, Dr. Mina Bissell, which says half of the secrets of the cell are actually outside of the cell, not inside. And I would actually go even further. I would say that half of those secrets are physical in origin and not, you know, just chemical, biochemical. So this is actually something that we are looking at in my group, how cells interact physically with the environment. So what kind of physical cues are we talking about? Now, what you are seeing here in the slide is on the left-hand side, you have a variety of physical variables that you can think of as inputs, you know, things like matrix density, substrate stiffness, ligand presentation, topography, and so on and so forth. So all of these inputs or variables have been shown in the past, I don't know, 20 years or so, to influence or to in some way affect the behavior of cells. And that is what is listed on the right as outputs, what I would call outputs. And the outputs can be cell phenotype, protein expression, differentiation, migration, and so on and so forth. So this is, in general, this is something that we are looking at. So how these physical inputs can shape, can drive the outputs from the cell, you know, the behavior of cells. Now, one thing that you will notice is that, you know, this kind of physical interactions, input outputs, is happening in all kind of situations. It is happening in all organs in the body. And it's also happening when you start to implant anything into the body. So the cells in the body will interact with the implants physically and also chemically, of course. So one way to think about this is it is through this bidirectional interaction between cells and the environment that our tissues and organs can actually function. And when something is wrong with this interaction, physical, mechanical interactions between cell and the matrix, and you have malfunction, which is basically disorders, diseases, and so on. So conceptually, this is what my group is looking at. And so what I thought might be a nice way to sort of, bring you through the activities in my group, I thought it's good to just take an example, just a very, very concrete example. And the example that I want to point out here is blood vessel. So, blood vessel is quite a, I mean, if you think about it, it is quite a simple context, right? So basically you have a tissue which is tubular in shape. So which means that, there is some curvature in the tissue. And within the tissue itself, the cells, for example, in the vessel wall, they have to interact with collagen fibers, also with elastic fibers and other ground substances. But let's focus on just collagen fibers. Now that by itself already provides vascular cells with two types of physical cues. One is the curvature of the tissue, and the second is the ligand distribution, which is from distribution of collagen fibers inside the tissue. So let's take this example, and I will sort of guide you through the two aspects that we are looking at. And one is curvature and one is ligand distribution. So the first, the curvature. So curvature is something about the geometry, right? So the geometry of the tissue. And if you think about it, pretty much all organs that we have in a body, they have some kind of geometry. I mean, they have to, right? So vessels, they have a tubular geometry, tubular shape. If you look at glands, for example, so glands have a more spherical ellipsoid kind of shape. So the cells have to, they are in that shape for a reason. So the shape itself can also influence cell behavior. If you start thinking about implants, scaffolds for tissue engineering, for example, then you're thinking about something like what I'm showing down here. So this is just a couple of examples of triply periodic scaffolds. And again, in this case, the cells have to interact with curved substrates. And we actually don't know a lot about how cells interact with curved substrates. So precisely because a lot of research has been done only on culture flask flat substrates, purely 2D substrates. So what we thought was a key challenge was to understand how cells recognize tissue and scaffold geometries. Now, and the geometries are quite diverse, right, as what you can see over here. So our strategy was to build a chip. So we microfabricated a chip, something like what you're seeing here, a chip containing basically a library of, a huge number of any kind of structures that you can think of, like cylinders, either concave or convex. So what I mean by convex is, hill shape, and concave is valley shape. So, convex, concave cylinders of varying diameters, varying sizes, ranging from micron size all the way up to millimeter scale. We also have a couple of, donut shapes. And the donut shapes are actually quite useful because mathematically they provide a Gaussian curvature that is quite distinct from cylinders because they are convex in one direction and concave in the other direction. But I wouldn't go too much into it. So we basically built this yeah, what we call curve chip, which is this chip containing a library of curved substrates for cells. So this is also what we call a 2.5D chip, meaning that it is not really 2D per se, because you have the structures coming out of it. But at the same time, it's more than it's less than 3D, right? It's not a real 3D situation. So we are presenting cells with 3D-like condition in a 2D chip. So that's why we call it a 2.5D chip. So we started basically putting cells on top of these chips and see what they do. And one quite striking observation was that if cells are sitting on top of a convex substrate, so in this case, it's human mesenchymal stromal cells. So these cells actually try to, they hug the structure nicely and tightly. On the other hand, if you start putting MSCs on concave structures like this, in some situations they can actually, so what my students like to call them is they do a Spider-Man. So they actually hang on to a few attachment points, a few adhesion complexes, and they can actually lift their cell body upwards. So they're not hugging onto the structure anymore. So that already tells us that somehow the curvature of the substrate can somehow influence the adhesion behavior of cells. So we did the next thing. So we started looking at how cells migrate on these substrates, so concave or convex. And what you see on these videos is, on convex substrate up here, the cells migrate in a very distinct way. So if you have seen a purely 2D cell migration, this is basically what they do. So it is a very typical 2D cell migration. On the other hand, if you put the cells on concave substrate, you have a totally very, very distinct migration move. So again, it is more like a 3D cell migration, more like, again, borrowing the analogy from Spider-Man, it's really like Spider-Man jumping from one building to the next. It's really 3D in behavior. So we started quantifying that, and we looked at the cell speed and cell direction. And one thing that I think is quite interesting to realize from cylindrical substrates, if you put cells on cylindrical substrate, is that depending on the direction of the cell, the direction of the migration of cells, they will see different curvatures. If the cells are elongated in the direction of the long axis of the cylinder, then they actually do not see any curvature. It's like a flat situation. On the other hand, if they're oriented in the circumferential direction of the cylinders, then they see the maximum curvature, essentially. So we basically hypothesized that depending on the direction or elongation of the cells, they will migrate in a different mode. So that's basically what we saw here. So when the cells are on convex cylinders, whenever they are elongated in the long axis of the cylinder, they migrate quickly. Whenever they're not, they migrate slower. And yeah, so on concave, it is a totally different behavior. So apparently, if the cells are migrating in the circumferential direction of the cylinder, they actually migrate faster. So we analyzed this further, and we actually saw kind of a universal relation between cell speed and the perceived curvature that the cells see. And apparently, the more convex the curvature is, the lesser the cell speed is. So it's kind of a universal relation that we see across all structures that we put the cells on. So all kind of diameters of cylinders, all kind of convex versus concave structures. So we thought this is really cool. So somehow cells can actually dynamically sense the geometrical cues from the substrate, in this case, in terms of curvature. And one thing to think about this is, all kind of organs in our body, they have some kind of curvature. So, this is a very nice figure that I took from the review paper from a group in Maastricht, and which is really summarizing the curvatures that you actually see in the body, in the eye, the heart, the kidneys, skin, bone, and so on. They're all curved, and not a lot of people actually realize that the curvature of the organ itself can actually influence cell behavior. And what is also even more important, I guess, is when you start implanting, biomedical devices into the body. It can be just a scaffold. It can be something more complex, cardiac patch and so on. The geometry of the implant itself can be used to shape cell behavior. So this is something that that is increasingly being recognized, and we should actually make use of this opportunity to guide cell behavior. Because at the same time, the fabrication technology has advanced so far until today. So we can actually build implants, we can build scaffolds with very, very precise geometry that we can actually use those for controlling cell behavior. So coming back to the example of Westall, so I've talked about curvature. We also looked at ligand distribution. And the way to think about ligand distribution is through a term that is called contact guidance. And yeah, so contact guidance is quite simple, essentially. So it's basically, the ability of cells to sense and respond to anisotropic cues. So basically, cues that have some kind of direction. So this has been shown for more than half a century now. I think it was first shown in the 50s. And I'm showing here a paper from the 70s that shows that when you put cells in in this case on a glass wire, the glass wire itself can actually drive the alignment of cells. So this is a very nice comparison between what cells do on anisotropic substrate. So basically substrates with micro grooves and micro ridges, as you can see here. And what the cell do if you put them on an isotropic substrate. So this is a very well-known phenomenon, but at the same time, it's quite poorly understood. People have started to realize that the same phenomena, contact guidance, is important and is playing a big role in a variety of processes in the body. So from developmental processes, morphogenesis, to cancer invasion, cancer migration, cancer cell migration, all of these processes are in one way or another influenced by contact guidance cues in the body. But we don't actually understand the mechanism behind contact guidance. So that's a question that a former PhD student that I guided asked herself. So what drives contact-mediated cell alignment? So the way that she approached this study is, so experimentally, basically she built purely 2D substrate, so without curvature, without anything, just a purely 2D substrate with micropattern proteins. So in this example, she created stripes of fibronectin, so an ECM protein, and the stripes have different widths and spacing between them. And in between the fibronectin stripes, you have a non-adhesive coating, so cells cannot really form adhesions on those areas. So when she put cells on these patterns, she saw, well, a variety of cell behaviors, right? So in some cases, cells align, in some cases not. And and that's quite normal. So if you work with cells, you know that cells always there's plenty of cell to cell variability. So you need to somehow take into account the statistics of it, so the statistics of the cell behavior. And that's actually what brought us to contact a collaborator, so Vikram Deshpande from Cambridge University, because they already had a statistical thermodynamics model for living cells. And that's actually quite a simple model. So if I just can explain it very briefly, they actually calculated the Gibbs free energy of cells by just a summation of all contribution of free energies. And the free energies are, again, very simple. There are two contributions, one from the cytoskeleton and one from the elastic, the passive elastic contribution of the cell itself. So that was their original model. And now we have a situation where there are spots, there are regions that cells can form adhesions, and there are spots where they cannot form adhesion. So we basically added one more element, one more contribution of free energy, which is free energy of adhesion. And it is, again, very, very simply modeled as a spring. So I'm not going to go into the detail and bother you too much with this. I will just jump right through the results. And basically what we saw was there are like two regimes of contact guidance. So there's contact guidance at large length scales, and there's contact guidance at small length scales. And again, I'm not going to bother you with the details, but we saw a lot of agreement between the experimental results and the very simple model that was built in the study, which is very encouraging. And what the comparison told us basically is that the two regimes of contact guidance can be explained by two different phenomena. So at the small length scales, it is really the gaps. So it's not really the width of the patterns that determines the contact guidance, but it's actually the size of the gaps between the patterns that drive this contact guidance. And at large length scales, it's interestingly enough, at large length scales, it is entropy that drives alignment. And why do we call that entropy? Well, because as you make the stripes of the fibronectin narrower and narrower, what basically happens is that you're restricting the orientational phase space that the cells can access, and that's an entropic penalty. So this is we found this to be quite interesting because in a way, this is a mechanism that doesn't rely on any kind of specific molecular mechanism, right? So this is a purely biophysical explanation for contact guidance. So I'm not saying that specific molecular mechanisms like the pathways that are always involved in focal adhesion formation and so on, they're not involved. So I'm not saying that. What I'm saying is that apparently contact guidance can already be explained through a purely biophysical explanation without needing to invoke any specific molecular mechanism. All right, so coming back to the example of blood vessel. So I've told you about curvature, I've told you about ligand distribution. And of course, I mean, if you're talking about blood vessel, you also need to think about the blood flow, right? So there's an effect of shear flow, and there's also an effect of circumferential stretch. And what do I mean by that? Well, if you take a look at an ultrasound of a blood vessel, so this is I'm going to play this video shortly. If you take a look at the ultrasound of the vessel wall, so what you see in red and green here are basically the outline of the vessel wall. you will see that the vessel wall is actually, expanding and constricting because of blood flow and because of the pressure from the heart pumping, as what you can see here. So basically, aside from the relatively passive cues, physical cues like curvature and ligand distribution, we also need to think about the more active cues, active mechanical cues like shear flow and stretch that the cells will experience in the vessel wall. So this is another project that we did. So we basically built a bioreactor, a vascular bioreactor that looks like this. So basically, each of these capsules is one bioreactor. And what you see, maybe it's a bit difficult to see, but what you see a bit whitish here, that's basically the tubular scaffold that we put in there to stimulate cells mechanically. So the nice thing about this vascular bioreactor that we built is that you can actually apply independent and spatial temporal control of each of these components. So the wall shear stress, the circumferential stretch, and we can also use the tubular scaffold in there. So we can actually, make use of what we know about the geometry, the curvature of the scaffold. Now, this, it may sound simple, but actually to be able to really decouple shear stress and mechanical stretch is it's a lot of work. It's not straightforward at all. So it actually took us something like 2 years to build a bioreactor that allows us to really decouple these two components. So, but what we were able to do with this is we can actually evaluate the effect of either shear alone or stretch alone or a combination to the the tissue regeneration in the vessel in the vascular construct. So this is just one example of that. So we were able to see that shear can actually reorient collagen fibers inside the vascular scaffold in the direction of the flow. Interestingly enough, the circumferential stretch can also reorient collagen fibers in the direction perpendicular to the direction of stretch. So this is quite interesting. And when you actually combine these two, shear and stretch, you basically accumulate the effects of shear and stretch. So you have a very narrow distribution of collagen fibers in the vascular grafts. Now, so we did the next step and we thought, well, if we build a vascular graft, for example, and we put the vascular graft in the body, so we implant them. So what happens is not only the physical cues like curvature, flow, stretch, it's not only that, but we also have to take into account the host response, right? So the immunological response. So that's where we teamed up with a group of Antal Smiths. And so what we did was, well, let's not just focus entirely on the tissue producing cells, but what if we also have monocytes? So monocytes and macrophages. So that's what we did. And so we basically co-cultured macrophages and tissue producing cells, the fibroblasts, in our vascular bioreactor. And we saw what kind of environment we are seeing in there. So what we saw was that a lot of anti-inflammatory cues, anti-inflammatory cytokines, for example, IL-10 over here, is actually being upregulated by stretch. So there's this jump over here. On the other hand, some pro-inflammatory cytokines like IL-6, MCP1, they are down-regulated by stretch. But what is also interesting is that this down-regulation of the pro-inflammatory cues are actually, in a way, counterbalanced by the shear stress. So you see that when you have shear stress, the regulation of IL-6 and MCP-1 comes back to near what it is in static condition. And when you have the combined stretch and shear stress, the combination is, it's not that, it's kind of a combination of the effect of stretch and shear stress. So the outcome of that is what is shown here. So apparently when you have cyclic stretch alone, you basically promote tissue growth. So you see a lot of collagen. So collagen is in green here. On the other hand, if you have shear stress alone, you don't see a lot of green. When you have combined cues, you have somewhere in between. So the amount of collagen is somewhere in between cyclic stretch and shear stress. So they're, in a way, the effect of these two mechanical cues are sort of balancing each other out. And that's what we also see in the eventual mechanical properties of the vascular constructs. So just to go back to the bird's eye view, we think that the physical and mechanical cues can be sensed and responded to by the immune cells and by the tissue-producing cells, both in a direct way. So both of these cells are mechanoresponsive, and an indirect way, meaning that the behavior of tissue-producing cells are responsive to the cues directly, but at the same time responsive to the inflammatory versus regenerative environment that is actually created by the immune cells. So right now we are actually exploring this further. So, just to very quickly sum up right now, the lesson learned is physics and mechanics matter in biology, and we can actually learn a lot from biophysical approaches and biomechanical approaches, or actually I should say mechanobiological approaches to understand processes in biology. And the physical environment of cells really induces distinct cell phenotypes in a length, scale, and dimension dependent manner. And there are a lot of fundamental questions that are still unanswered, and that's what we are trying to go into. But at the same time, I would also like to think of this as a tool. So we can actually, if we know how cells respond to these passive and active cues from the environment, we can actually devise ways to instruct cell behavior. For example, in tissue engineering or building of biomedical devices. And as you realize, to think about it in this way, you need to think about this in a very multidisciplinary way. So you have to have expertise from biophysics, mechanics, but also synthetic biology, biochemistry, and so on. And that's where that's basically how I built my team. So if you look into what my team is working on, so we combine expertise from a variety of disciplines to approach these problems. So what I thought I would also say just very, very quickly is just to give a glimpse of what kind of projects are kind of being undergone right now in my group. So we are looking into the combination of cues. So I've shown you before about curvature and ligand distribution. We are trying now to see if we can actually combine curvature and ligand distribution and see, like, is there any competition between these two cues, or are we seeing synergistic effects? And the context of this is we are now looking at the corneal stroma in the blood vessel, because these two tissues are curved, and they also have very, very specific tissue organization that are very important for the function. If you look into the corneal stroma, it has to be transparent, for light to go in. And the transparency of cornea is really dependent on the structure of the collagen inside. We are also looking into the effect of combined effect of, for example, contact guidance and strain. and in this case, in the context of the heart, so the beating of the heart, and how can we, devise a way to restore tissue organization after myocardial infarction. In a more fundamental way, we also want to understand, we want to get some more fundamental insights into how cells can sense these physical cues from the environment. So both in 2D, 2.5D, and 3D. So those are what is happening in a single cell level. We are also looking at multicellular and tissue level. And a couple of examples are here. So we are trying to see if we can think of a smart way, a clever way to build vascular grafts that when you implant these grafts into the body, we can actually instruct cells to form tissues that resemble native vessel walls. So that's something that I'm doing together with Miguel Castillo. We are looking into polarization of epithelial monolayer together with Patricia Dankers. Another tissue that we are looking at is the blood-brain barrier. So we are looking into how we can actually use physical cues to microengineer blood-brain barrier. Another subject that we are starting to pursue is the nervous system. So we are trying to see if we can, again, using physical cues from the environment, whether we can actually guide the formation and maturation of tissues and organoids, and eventually whether we can actually tune the neuronal interconnectivity in developing brain tissues. So I would actually like to stop there and acknowledge basically my team, which is boxed right here. So a couple of people have already left, so they graduated with their PhDs. But I think I already mentioned a number of names in the previous slides, but this is basically my team. But I wouldn't have done, I wouldn't have been able to do those kind of works without the collaboration of these people on a variety of topics like cell modeling, fabrication of materials, curvature, soft matter, image analysis, and on a number of different tissues. And with that, I would be happy to discuss further and answer your questions. Thank you very much.
Well, thank you so much, Nicholas. Let me just fix this here. Thank you so much. I think a very fascinating, very clear presentation. And I think what you've provided is a lot of opportunity to explore possibilities for collaboration, because it seems, especially when you are giving an outlook at the research topics that you are starting to look into, there's presumably opportunity to join forces with researchers at other institutions. And we have a couple of questions already that have come in, and one of them is Andrew. Andrew, if you would like to join us and ask your question.
Yeah, sure. Can you hear me?
Yes, we can.
Okay, it won't actually let me turn my video on, but that's my face, so just imagine it talking. So Nicholas, it was very cool to hear you talk. Really beautiful work. I think it's super exciting moving forward how you're going to start combining some of these cues together in single systems to start to disentangle some of these inputs. It's really fascinating stuff. So one thing I was just a little curious about with the micro patterning you were doing on these sort of 1-by-1 stripes where the cells can spread really nicely, but you do still find that they align really well along the direction of the stripes. I'm wondering if you could talk a little bit about the focal adhesion, elongation, or alignment in these systems, and what might happen if you can knock down some subset of focal adhesion proteins, prevent them from making long directional adhesions, whether you would still see cellular alignment. So the question essentially is, how much does the focal adhesion alignment drive cellular alignment?
Yeah, this is a very interesting question that we also try to answer ourselves when we wrote the paper. So I know that there's a couple of papers coming out from which group? I don't remember which group it was, but there's a couple of papers that basically um, reports or actually showed or demonstrate that the formation, that the elongation of focal adhesions is basically what is very important for driving the entire cell elongation and alignment. Now, so what we tried to do in this study was, as you go from large stripes into narrower and narrower stripes, you will basically hit a point when the width of the stripe is too small for a full elongation of focal adhesion. So we basically went down to, I think, 1 or 2 microns in width of the stripes. And the focal adhesions, when they form and when they're fully mature, they can elongate up to 5-6 microns, especially for the cell types that we are seeing, that we are using in this study. So in those situations, basically what we see is that the focal adhesions can only elongate in the direction of the stripes, right? So they cannot really elongate in the transverse direction. So they cannot sort of bridge between two.
Yep, yep.
So what happens there is we are forcing the elongation of the focal adhesions along the stripes, and we are seeing the correlation with the whole cell elongation and alignment. Now that is, I would say that's a correlation and not so much a causation, because we also see that when in situations where the focal adhesions can still fully elongate in all kind of directions. So when the stripe width is still broader than the length of the fully mature focal adhesions, which means that the focal adhesions orientational distribution is still very random, we still see that the cells can elongate and align mostly in the direction of the stripes.
Mm-hmm.
Basically, that tells us that it's more a correlation rather than a causation. Sure. And elongation and orientation of single focal adhesions within the cells is not enough to drive the elongation of the whole cell.
Sure.
So that's what we are seeing.
Oh, very cool. So like on these wider ones that you're mentioning, you kind of showed in the diagram, I think the model diagram, that on the outer edge where the cells might be kind of interacting with the actual boundary itself.
Yeah.
You have some strong alignment, but then in the center, you kind of showed that they were free to go anywhere.
Exactly.
Is that what you also saw experimentally, or did you see some sort of subset of cells that aligned really well, even though they weren't interacting directly with the interface between the patterned and unpatterned areas?
This is also a very interesting observation that we observe, because if that is the whole story, then basically if the width of the stripe is twice or 3 times the cell size, then you wouldn't see this effect, right? So you would expect that the cell orientation is still all over the place and they can still fully elongate in all directions. But that's not what we saw actually. So somehow, even when the stripe width is like 5 to 10 times cell size, the cells can still, if you look at the distribution of cell orientation, there's still a strong alignment of cells in the direction of the stripes.
Wow.
Now, this, why I think this is interesting is that because it seems like there is kind of a history. So the cells have a history when they already touch the edge and they're somehow forced to align in the direction of the stripes. Yeah. Even if they migrate towards the center, they still have that history. So they're more likely to still align in the direction of the long axis of the stripes.
Awesome. Thank you, Nicholas. Nice to meet you. Nice talk.
Nice to meet you.
Thank you so much, Andrew. Now, do we have any other questions? If anyone has any questions now to ask to Nicholas, please raise your hand and we will allow you to switch on your mic and your camera. The question doesn't necessarily have to be a scientific one. If you're just interested, for example, as I am, Nicholas, to learn about opportunities to participate in your lab, maybe you can share a few words about, for example, the PhDs that you offer or other opportunities that you offer for researchers from the region to work with you.
Yes, so this is, I think, where the ERC comes into play. So yeah, 2 years ago, I obtained my ERC starting grant. And so the information that I also got from EURAXESS, in fact, is that there's an opportunity uh, for non-EU researchers to actually come and visit, for a research visit to the labs of the ERC laureates. And this will be partially financially supported by the European Commission. So I'm open to possibilities. So I kind of already sign up for the possibility for the program to European Commission. And if there's anyone who thinks that, you have the expertise to complement what we have in the lab, if you're interested in what we are doing, just drop me an email and we can always think of something.
Excellent. I think that's a very positive message. And how would anyone who's interested get in touch with you? What's the best way to find out more about your research and your research group? Do you have a particular website?
Yeah, so I have to say that the website of the university where I'm working is not that great. It's kind of complicated to navigate. So I guess the easiest way is just to email me. So drop me an email, and we can always set up an online meeting, Zoom or Teams or something, just to get to know each other a bit better.
Excellent. Thank you so much. So that's a great message from Nicholas. Now, I think we had another raised hand here. Just looking who the person is. There was a Christine who had a question. Is Christine still with us now? Let's just see.
We have Gerald. Susan?
Gerald. There's Gerald. Please,
Yeah.
Gerald.
Oh, sorry. Good afternoon to everyone. Mind if I just turn off the camera? I would like just to ask Dr. Kurniawan. May I ask, sir, what to a layman will be the benefits of your research to everyone? Of course, I have been following your research. I mean, through this Zoom seminar, but I know for us in the ground level, I mean in the grassroots down to the people, what will be the benefits of this of your study? I mean, what will the benefits of your research to everyone? I mean, because not all will understand. Thank you very much.
Yeah. So basically, if I can sort of rephrase the question, so how can we actually translate the fundamental understandings that we are obtaining from our research to something that the public can benefit from, right? And yeah, maybe I can just give a couple of examples. So our efforts in the vascular grafts, right now we are trying to kind of develop or think about the best way. Is there any kind of ideal grafts that we can develop that can actually instruct when you start implanting those grafts in the body, it shouldn't invoke some kind of a host response. It shouldn't develop neointimal hyperplasia, which is a very, very common issue with vascular grafts. So how can we prevent that using smart designs of the grafts, using smart coding, smart geometry of the grafts? So these are actually something that we are collaborating together with a university in Utrecht in the Netherlands. So we are basically developing the fundamental aspect, and they're working on the animal studies. And together, we are trying to see if the grafts that we come up with can make it into human. So one sort of pre-product that we have been able to work with so far, it's I would say it's close to first in man. So I guess that's a nice progress. Another example that I can also mention is our work with brain tissues. So we are developing basically in vitro brain tissues in the lab. So starting from iPSC, so induced pluripotent stem cells. So we are developing brain tissues on chip basically. And so far the problem with brain organoids and any kind of in vitro models that are out there for the brain is that it's quite difficult to evaluate the 3D neuronal interconnectivity. So it's you basically, with those models, you have to basically disperse the cells again in 2D and then evaluate the interconnectivity in I would say, relatively artificial environment. So we basically, what we do in our lab is we build a completely different way of building a brain tissue from scratch from iPSCs, and which actually enables us to evaluate and monitor the 3D interconnectivity of the neurons. Now, why do we think this is interesting is that because if you can actually do that, then you can study things like propagation of abnormal discharges. So which is the basis, the clinical basis of epilepsy, for example. So we have actually achieved that. So we can actually do kind of epilepsy on chip. So we can actually recreate the propagation of abnormal discharges from neurons. And the nice thing, the cool thing about this is that now you can really test a variety of treatments, right? So you can do kind of a drug screening on this platform and see which drugs actually prevents the propagation of abnormal discharges. Or you can also think about like, I think an emerging treatment nowadays for epilepsy is the deep brain stimulation. And of course, the problem with that is you actually don't really know what is happening to the neuron level when you do a deep brain stimulation. Now, what we are trying to do now is, can we actually do deep brain stimulation, or can we simulate deep brain stimulation in our chip, in our brain tissue in chip? Now, if we can actually do that, then maybe we can tune, we can modulate the way that we stimulate the neurons in the brain using our in vitro setup, which can then be translated to more clinical setting. So I guess this couple of examples can help answer the question.
Thank you very much, sir. Thank you very much.
Thank you, Gerald, for the question, and thank you, Nicholas, for the wonderful explanation. Now we have another question, maybe our final question, looking at the time. The question is from Custer. Now, if you want to switch on your microphone and video.
Oh, hello. I'm Custer from Philippines. I'm with the Philippine Nuclear Research Institute, and we just started our work with scaffolds a few years ago, and we're developing kidney scaffolds, bioscaffolds by decellularization. So here's my question. It's a fantastic and amazing that information can be derived from the shape of the extracellular environment. So uh, would you suggest to have bioscaffolds because of the complexity rather than coming up with synthetic polymers for regenerative medicine? And that's my first question. And then a follow-up question is, uh, what about I saw this company called Helionics, and they develop polymers depending on the pore geometry and the size, and without the inflammatory response by the mesenchyme, I see the stromal cells, but somehow with your model wherein you mixed uh, the immune cells with I don't know the fibroblast, there was an inflammatory response. Is there a way we can manipulate inflammatory response merely by the contact guidance mechanism and the curvature or the shape of the pores or of the surface?
Yeah, this is actually a complex question. So, in fact, I think about 6 months ago, so about half a year ago, I attended a conference, and there was an open panel discussion exactly about this. Like, I'm referring to the first question about what kind of materials, synthetic, natural, decellularized matrix, and so on. I don't think we can actually resolve this problem in 5-10 minutes. I mean, we had a discussion of like 3 hours back then, and and we did not come up with a satisfactory solution or satisfactory answer. So it's a complex question, and I don't think there's a straightforward answer either. So I think, I mean, it's just my opinion, but each of the approaches has their own pros and cons. So maybe the way to go is really kind of a hybrid approach. So you have to make use of the strength of one approach, like the tunability of the synthetic systems. At the same time, you have to borrow the strengths of the natural systems and tune the ability of uh, uh, tune the cell adhesion properties. Yeah, I don't think there's a straightforward answer to that. So, I mean, I'm happy to discuss more about this. And the second question, I actually forgot. Can you repeat the second question again?
You're muted right now. You need to unmute yourself.
It's about Helionics. It's a biotech company where they develop polymers and then they're able to come up with some healing properties without the inflammatory effects. So, in your experiment where you mixed immune cells, is there somehow a trend in terms of the pore size or the contact distance
Right.
and the inflammatory reaction?
Yeah. So this is I cannot answer that. So the quick answer is I don't know. The longer answer is it's something that we are actually doing right now. So in the first couple of studies that we already published, we did not actually vary the scaffold condition. So what we vary was, the ratio between the tissue producing cells and the immune cells, the amount of shear flow. So really kind of a more proof of concept studies, if you like. So what we are doing right now is we are really changing the scaffold parameters. So exactly what you mentioned, like what if we change the pore size? What if we change the microstructure of the scaffold? Smaller, bigger diameters, or more straight and or more wavy fibers? Would that have any effect on the response? So that's actually what we are doing now. So I hope that if we do this kind of a webinar again in a year or two, hopefully I have the answer, but not now.
Thank you, Dr. Nicholas.
Thank you so much. We had a wonderful presentation. Nicholas, really, that was fabulous. Even for me as a political scientist, I was able to follow what you were talking about, and it sounds absolutely fascinating, and especially how your research is applied and what benefits it has for people, for humankind. I think that is fabulous. So thank you so much for your work, and thank you so much for taking the time to share your work with all of us. And I think to all the attendees, as Nicholas has said, he is open to be approached by you. When you have a question about his work, I think you can contact him. And as he said, there are opportunities, of course, for you to join forces with him. So if you think your profile fits with the work that he has described today, then I would also encourage you to reach out to him.
Well, one thing that I can add is, the corona situation doesn't make things easier. So at the moment, the governmental restriction and university restriction doesn't actually allow mobility, a lot of mobility. So I hope that the corona situation will get better soon. If so, then we can sort of open up our doors again. But at the moment, unfortunately, that's a bit limited.
Absolutely, that's what it is. So everybody, of course, stick to the restrictions. Try to get a vaccine if you can, so that we all get out of this mess as quickly and safely as possible. Now, I'd like to just, before we close, hand over to my colleague, Dr. Jenny Elmaco, and she also would like to say a few words.
Yes, thank you, Susan. Thank you so much, Dr. Nicholas, for that. I took up biology for 2 years and somehow shifted also to the social sciences. But listening to you, I mean, there's a lot of secrets to the universe, and hopefully, really, we can get together and collaborate more, even during this time of this pandemic, and we can build stronger, post pandemic. We have a little surprise for you. If I can ask Ben to show the screen. Ben. So this is just our small token of appreciation. So we have a special certificate for you. If Ben can show it right now. While he's doing that, because I still can't see it, for those of you who need a proof that you attended this webinar, we will be in touch with you through an email after this event. And also, this event with Dr. Nicholas is not just we don't stop here. We have a series of webinars that are happening in the next days. Tomorrow, for example, we have a seminar on IP in research, and this is with the European Union IP Help Desk. So if you'd like to come on board and join us in that discussion, please do so. Next week, we have a series of webinars, info sessions on the Marie Curie Skłodowska actions. So we have one on the 22nd for those who want to do a doctorate in Europe. And we also have on the 20th and on the 21st for those who are already have their doctorates and want to do a postdoc. And on the 28th, for those of you who are representing institutions and you'd like to host a global fellow, and this global fellow has will be coming in with funding from the Marie Curie Global Fellowship Scheme, then please join us also on the 28th. So please follow our social media accounts on EURAXESS ASEAN so that we can continue this partnership, and then we can help you as well as researchers as you sharpen your skills in research and also identify areas of collaboration. So I'm still, Ben, I can't see the screen, unfortunately. Do we have some problems with showing the screen, Ben?
Yes, we do.
There you go. Okay, now I see it. So, so Dr. Nicholas Kurniawan, please, we are humbly giving you our certificate of appreciation virtually because of the pandemic. But thank you so much for sharing your knowledge and expertise. And this is given during the Meet My Lab event on the 14th of April, 2021. And that is, of course, signed by EURAXESS ASEAN and the European Union as the funder of this program. So thank you so very much for that.
I appreciate it. Thank you so much. It's a pleasure to be invited to this event. Thank you.
Thank you so much.
Thank you so much, Nicholas. Thank you so much, everyone, for joining, and everybody stay safe and have a wonderful afternoon, rest of the day. And we'll see you soon. The Meet My Lab series continues, of course. We have sessions set up, the next one in June on cosmology. And we also just secured a speaker or a team of speakers on artificial intelligence and music. So really very exciting topics coming your way. Do make sure you follow us on our social media accounts, on our website. And we're also now on Spotify. So if you haven't had enough of Nicholas' talk today, you know, you can listen to it all again when you're in the shower or in the car or wherever you are. And learn even more. So once again, thank you so much, and we'll see or hear you all very soon.
Thank you, everybody. Take care. Bye, Nicholas.